All right, guys, how you doing? Welcome to 3.4, the fourth and final section of this chapter. Today, we're going to look at stem cells. So let's jump into it. Okay, while most cells in an organism have the same amount of DNA, that is, your brain cell will have all of the genes and the DNA to make up your whole body, and so does your heart cell and your muscle cells. Many of those cells are specifically suited to a particular role, right? The cells, when they develop into these specialized roles, we call that process differentiation or specialization, right? Differentiation is often permanent, which means that when the cells become that type of cells, they will develop those particular traits and lose the traits to do other things. So, for example, when a cell becomes a brain cell, it no longer works as a heart cell because it only has that particular role, that particular specialization. It will switch off the genes to make it into a heart cell and keep the genes that make it into a brain cell on, right? Now, when cells haven't differentiated or become specialized, we call them stem cells, right? So stem cells are undifferentiated cells that haven't become or taken on any particular roles in the body, right? So here's a diagram, and you can see we start with the zygote, and then as you go down, they become different types of cells, and then those particular types of cells will then differentiate to become different types of different parts of the body. So, for example, the mesenchymal stem cells will soon become the bone or the cartilage or the muscle or the marrow. And then you've got hematopoietic stem cells that could become either red blood cells, white blood cells, or platelets and so forth. And then the nerve cells can become different types of nerve cells, right? And stem cells can be often thought of like students in society, right? So students, you guys haven't made up your mind as to what your career is going to be, and you haven't specialized into a particular role or any particular functions in society. You are currently undifferentiated, and you are like a stem cell. Okay. Now, the stem cells often revolved in two particular processes. The first is differentiation, as I mentioned. The second one is actually what we call self-renewal, replacing itself as a stem cell, and I'll explore what that looks like later. All right, so here are a few examples of stem cells. You got your hematopoietic stem cells that come from your bone marrow, right? Human blood contains 4 substances. You got your plasma, platelets, white blood cells, and red blood cells. Now, aside from the plasma, which contains all your nutrients and waste, the other 3 are cells that used to be hematopoietic stem cells, right? And the hematopoietic stem cells in your bone marrow will divide, and then one of those 2 hematopoietic stem cells will become the original stem cell to replace itself. The other one would then transform and slowly turn into a red blood cell or a white blood cell or a platelet. Another example are basal stem cells in the epidermis. So, for example, humans lose about 30 to 40,000 skin cells per hour. I mean, that's a pretty high amount, which means new skin cells need to be produced at a fairly rapid rate. And it occurs in this layer below your epidermis called the basal layer. And there's your basal layer right there. And those cells will do the same thing, right? They will divide into 2. One of them will replace the original one that just divided, and the other one will go on to turn into skin cells, which then become the epidermal layer, the skin that protects you from all the other stuff, okay? Now, when we think of stem cells, we often have to think about the level of potency that the stem cells have. Potency refers to its ability to differentiate into other cell types. The greater the potency, the more capable that stem cell is, right? The greatest level of potency is what we call a totipotent stem cell, which can turn into any cell type in the body. It doesn't matter whether it's a heart cell or a brain cell or whatever. It has the ability to turn into those stem cells, right? And so the only cell in the body that can do that are actually the zygote, up until when it turns into a 16-cell large kind of clump of cells, right? So you got your zygote, which is the first cell, and it's going to turn into 2, 2 into 4, 4 into 8, 8 into 16. After 16, it begins to split off between turning into the embryo and turning into the placenta, right? So up until that stage, it is what we call totipotent. After it turns into either the embryo or the placenta, whatever turns into the embryo can't become the placenta, and vice versa. Here's a diagram of your zygote, and here's it dividing and turning into an 8 to 16 cell clump, right? Now, after a totipotent stem cells, it is what we call pluripotent, which can form a range of cell types, but not all, right? So the cells of the blastocyst, which has now differentiated between the embryo and the placenta, will form what we call pluripotent because they can turn into any of those germ layers in the body, right? So that particular process, that's called gastrulation, and it can turn into one of three different layers, right? It can become either the ectoderm, which is like your skeleton and your brain and so forth, the mesoderm, which is your heart and your kidneys, or the endoderm, which could be things like your gastrointestinal tract or your lungs and so forth. Okay, so once these germ layers form, the cells are no longer pluripotent. So up until that stage, between 16 cells and about 64 or ish cells, they are pluripotent, right? They can become any of those germ layers, but they can't become the placenta, that's why it's kind of pluripotent and not totipotent, right? So these are the typical cells that are drawn from embryonic stem cells, which we will talk about later in terms of location. Okay. From there, pluripotent becomes multipotent. So any one of those germ layers are multipotent stem cells, right? They are, they will turn into specific types of tissues and organs. And the adult stem cells that we obtain our stem cells from are considered as multipotent, right? From multipotent, you have oligopotent, which can only form into a few blood types. They are also considered as progenitor cells, and a good example is the lymphoid cells, which can become either T cells or B cells, both of which are immune cells, but they are different. These lymphoid cells can become either one, but they can't become red blood cells because the red blood cells are from a slightly more kind of higher branch, right? After oligopotent, you have unipotent. Uni meaning singular, which means they can only turn into one type of cell, right? And the unipotent stem cells are often considered as precursor cells because they can only turn into that one type of cell, right? So a good example is the basal stem cells that I talked about earlier, which can only become skin cells and not like other things on the skin, like the nerve, for example. Now, let's talk a little bit about where we get our stem cells from, right? So our stem cells come from two sources, either embryos or adult stem cells, right? Embryonic stem cells are cells that are obtained from the inner mass of the blastocyst, which eventually forms the embryo, right? These cells are pluripotent. They can become any part of the embryo, right? As opposed to the adult stem cells, which are multipotent. We usually get these embryonic stem cells from embryos that were developed in IVF, and subsequently we kind of obtain these stem cells, which eventually destroys the embryo. So you can see a few ethical questions being raised there. Here is a small little kind of animation of how IVF works. That artificial insemination process is kind of, obtaining the egg from the ovaries and the sperm from the testicles. And then we actually put them into a petri dish and we artificially inseminate them to join them together and turn them into a zygote. And then we transfer them into the female uterus to grow them into a baby. Now, this kind of artificial insemination process develops a lot of embryos because you're trying to get the highest success rate as possible, but not all of them turn into babies. So the embryos that are no longer used usually get discarded if they're not going to be used for anything else. So we usually obtain embryonic stem cells from them, and we use those embryonic stem cells for a whole range of different purposes, which fall under this particular branch of medicine called regenerative medicine. The idea to regrow, repair, and replace damaged cells using embryonic stem cells, right? It includes a type of therapy called stem cell therapy, using stem cells to treat or prevent diseases and conditions, right? Currently, the only stem cell therapy that is approved is bone marrow transplant, which is a little bit more broad and easy and more regulated. However, if it is possible, you can think about the applications being very broad. Imagine having a type of cell that can turn into any part of the body, reinserted to heal or repair cells that were once damaged by the body. Things that we thought were permanent injuries could be reversible. For example, neurodegenerative diseases like Parkinson's, right? Or Alzheimer's or MS. Any sort of damage to brain and spinal cord, heart diseases, blindness, and even things like diabetes and pancreatic beta cells that we have, right? So you can see there the range of applications is very broad, but then again also some ethical questions we need to think about as well. The second category of sources for stem cells are the adult stem cells or somatic stem cells, and they are obtained from adult tissues in the placenta or cord blood of newborns after they are born in the umbilical cord, right? So these cells are either multipotent or unipotent, right? They're easier to obtain than embryonic stem cells and therefore have less ethical objections, but their capacity to differentiate is more limited, and you can't use them for as many things. Like, for example, neurodegenerative diseases aren't usually, you can't tackle that with adult stem cells. But you can see that, from all the different sources, from teeth, blood vessels, liver, bone marrow, and so forth, all obtaining these adult stem cells. Now, we can't talk about embryonic stem cells and adult stem cells without thinking about the ethical considerations, right? So I want you to kind of hit pause on the video and just have a think about this particular question, right? What are the key ethical considerations for embryonic stem cells and adult stem cells? You know, what are reasons to be for ESCs and adult stem cells, and what are reasons to be against? All right, I am going to explain it in 3, 2, 1. Here are the ethical considerations you need to think of, right? Concerns around embryonic stem cells include things like destruction of the embryo, maybe the idea of getting embryonic stem cells from embryos and things like that might lead to increased rates of illegal abortions or any sort of dodgy dealings and things like that. So there's lack of legalities and clarification of rules and possible commercial exploitation of obtaining stem cells for medicinal applications. Other things that might be for it could include things like, obtaining the stem cells come from donated frozen embryos that would have otherwise been discarded. In other words, we were going to throw these stem cells out anyway, so we might as well use them to actually save somebody else's life. Other things could also revolve around consent being a little bit more easier if we are talking about donor and recipient, and then also you can regulate any sort of trade or compliance guidelines and supervising any sort of banks that we might have to tightly regulate and stop it from turning into something dodgy and illegal. Considerations for adult stem cells, things that can concern or are of concern could mean opening the door for things like human cloning, any sort of breach or genetic manipulation like designer babies and that idea, and also perhaps the lack of legalities and rules and commercial exploitation. However, there are positive sides, right? Things that are maybe good could be the very obvious, the avoiding the embryonic stem cell pathway and therefore making it much easier for us to obtain them with less ethical kind of gray areas. Once again, you can set up compliance, you can set up supervision, you can set up oversight committees and so forth, and then you can also set up trade prohibitions to stop people from doing things like trading people for harvesting organs or stem cells or whatever, right? So those are just some key ethical considerations. Now, it probably is also worth mentioning that, one of the more kind of recent medical endeavors, and when I say recent, I mean the last kind of 15 or so years, is actually induced pluripotent stem cells. These are stem cells that are developed by actually reprogramming the adult stem cells that are no longer pluripotent to become pluripotent again. The idea was developed by Shinya Yamanaka in 2006 on mice, where he used these transcription factors to actually reprogram the genes that were switched off in the nucleus and essentially switch them back on so that the cell has the ability to be pluripotent once again. This also means that in terms of application, you take the cells from the same person, you're reprogramming them, maybe cultivate them en masse, and then you reinsert them back into the same person so that the cells can actually perform those particular applications for healing and recovery and so forth, right? So there's no immune rejection. You're not using somebody else's genes or somebody else's cells, and it doesn't involve any sort of embryos. So imagine that if you have a neurodegenerative disease, a doctor could grab some of your skin cells, reprogramming them to become pluripotent again, reinsert them back into your brain or your nerves, and then allow that healing to occur in your nerves. Once again, some really breakthrough stuff, but once again, kind of early days, still not fully, 100% just yet. So there you go, guys. Hope you guys enjoyed that and you were fascinated by some of the interesting things about stem cells. That concludes this particular section and this particular chapter, and I'll see you guys in chapter 4. See ya.